Claire Steves from the King's College. She's Professor of Aging and Health and Geriatrician and Clinical Director of the Twins UK cohort. And today she will speak about biomarkers and healthspan insight from a longitudinal cohort of adult twins. Thank you.
Thank you so much. I'm so honored to be here, and thank you very much to Laura for inviting me and to the Fondazione, because it's such an incredibly inspiring initiative, this, bringing together folks to study healthy aging. And I've been really enjoying this morning, and lots of thoughts for the panel maybe later. But I want to talk to you a little bit about some of the work that we've done within King's College London with a national cohort of older adult twins, where we've been studying a lot of the things that we've been talking about already today, and hopefully I can share some of them with you. Now, I just need to make sure the thing... Right, so many of us might be interested in intra-individual aging resilience, and the reason for this is quite clear. I wonder whether you can see that this picture of me is not from now. This is actually from 5 years ago, and you can maybe think about which version of the future me I might represent. But basically what I did was I put that picture into an AI image and asked it to age myself, and I got the top one out, and then I asked it to do it again, and I got the bottom one out. So we can imagine that there are two versions of me. One has aged a little more healthily, and one has aged a little less healthily. So then the question is, does that matter? Well, it might matter if something happens to me. For example, I trip on a pavement that's uneven and I fall, because the younger version of me might have enough time to put the hand out to sort of like hold the fall, maybe might fracture the wrist, but then might get over the wrist fracture, fairly minor injury, and go back home and live a very good life. But the other version of me might have some degree of impairment of reaction time or maybe muscle strength, and then may have a fracture of the neck of femur and go into hospital, and a series of events might happen which then lead to problems. And so this aging resilience in individuals is really important to consider. And I've got three sort of like areas where I think we can really think about this. The first is, what can we do to predict the future, so work out which one of these might be happening to me, but also importantly then change the future. What can we do to change that future for the better for healthy aging? The second is, how can we actually prevent and lessen the stresses? So we talked about that trip in the road. You know, these are environmental factors that as a community we can come together to reduce the stresses upon older people. And the third thing is that what can we do to really help recovery? And of course, as a geriatrician, that's what I do in my clinical work, but actually as a researcher, what I really want to do, and what all of us, I think, want to do, Davide, is put ourselves out of a job. We don't really want to have to exist in the future. So that's about intra-individual aging, but of course it's not all about individuals, it's about everybody, and ultimately it's about everybody living most of their life in great health. And here I'm going to show you, hopefully, if it will work. This is a sort of diagram. Now, the two bottom graphs here are actually real data, and you'll notice there are no confidence intervals on them, because this is the whole population of the UK. And what I've done here is I've taken people who've answered the census in 2021, and I've plotted them, the people that are male and who are in the highest sort of index of multiple deprivation. So they, in other words, they are not deprived. They're 10 in the group, so they're affluent people who are male, versus the females who come from the lowest socioeconomic area deprivation level. And you can see that there's a really big difference. And then what I've done there is I've plotted an ideal and aspirational one, which might be the yellow one, where maybe in the future we could get to a point where everybody is in very good health. But the point is that there's a difference, and we know that, between different people. And I think what that tells me is that we've got a lot to do, but it's also that potentially there are things that we can modify. There is potential for change here. It isn't inevitable. And of course, this is the health span gap. And I'm deliberately talking here about people who are living in very good health. So this is the sort of pinnacle of, in a sense, healthy life, rather than necessarily thinking about diseases which might come in there as well. But the reason why I sort of talk about this is as well, I think we need to think about what we're trying to do in the long term very carefully with aging science. And this is because there might be some that might think that what we need to deal with is this transgenerational gap. So in other words, to live forever. But in my view, I think the important thing is that we do. It is a natural and important thing that we are only ephemeral upon this earth. And the reason why is because we want to leave room for others and new generations coming forward. So the ideal is to have the longest healthy life, but then we do die. So on this note, I wanted to talk a little bit about age versus aging and biomarkers and geroscience. And I wanted to sort of bring to you this idea about a sphere of different measures, as it were. And I think we often think in 2D because it's easier in a paper to draw a graph, which, draw a graph which shows one single dimension. But the reality of aging is that it is multidimensional. And we've got to question whether or not there's a single biomarker of this complex 3D structure. I think it's really important to think of aging as a sphere like this. And ultimately, the size of that sphere is what WHO is defining as this functional ability, enabling well-being in older age. So it's that kind of volume of the whole space. And what Davide was just speaking about in multimorbidity, if you can imagine that these axes coming from the middle are different physiological states, in diseases what happens is we get certain dents upon that three-dimensional structure. And as Davide says, these are clustered together in big groupings, which then influence the function. Am I okay?
Yes.
Speaking
You're
too
perfect.
fast?
Yes.
Too fast, yes, too fast.
This is the problem with translation.
Yes, of course. So I will speak slower. So the idea is that each individual pathway going from the center of this sphere is an aging biomarker or a physiological process. Multimorbidity is the continuous dents of disease upon that three-dimensional structure. And so when we come to try to understand this in research studies, we are generally using markers, and some of them, like grip strength, can capture a little bit more of the volume of that sphere because they're measuring more than one aspect. They're measuring both sort of biological function of muscles and nerves, but also psychological function and physical function together. But when we look at a biomarker like that, we need to be careful we're not looking just at age, but at aging. And what I mean by this is that instead of studying the difference between young and old, I really would encourage us to study the difference between resilient and non-resilient in the same age. And indeed, we need to not just look at one of those dots, but we need to look at how that dot changes over time. And that's what I've been trying to do with our twin studies. So I'm just going to go through this a little bit more just to reiterate for you about this conceptualization of how we might think about biomarkers. Because we might think about those individual brown lines there, which are markers of specific organ function or specific physiological function. And they might be underpinned and related to different biological pathways. So other speakers this morning have already talked about, for example, inflammation pathways. But within inflammation, there's a lot of different specific pathways which might then influence multiple traits. But ultimately, the volume of that balloon or that sphere is what we can capture in some of these measures of whole system functioning. And so on the right-hand side here, I've showed the hallmarks of aging, which we've seen already. And those things are mapping these biological pathways. But also the whole system functioning might be mapped more by other measures, which are similar to the measures in intrinsic capacity, which is this new WHO-derived method of measuring aging and vitality in older age. But also you can have biological common markers which mark overall functioning of a system. So on Tuesday, I was speaking in a conference in London Healthy Aging Network with Steve Hallvarth, and we were discussing how epigenetic clocks, actually, they're capturing a wide range of different insults on aging pathways, and they therefore are sort of, in a sense, integrative biological measures of whole system functioning. And the same might be made of other markers of biological aging, like, for example, telomeres that we talked about earlier on today. So we've been looking at these, as I say, in a longitudinal study of twins. Hopefully I will be able to... Oh, yes, no, what I wanted to talk about here is this little sort of tap on the end. And ultimately, in the process of aging, what happens is there's a leakage from this balloon. Now, it's not quite like we let off a balloon in here and it would just go very quickly out. It's happening very slowly over a lifetime. But then, and the corollary is, what can we be doing, actually, to be pumping up this balloon? And there are certain overarching mechanisms which I think can do that. And that's what's at the bottom of the geroscience hypothesis. So in a sense, what we want to be looking for is markers that can increase multiple different systems of functioning in aging, rather than ones that necessarily only tackle single systems, which, as Patricia was talking about today, some of the drugs might be doing. We need to be thinking about more holistic approaches. So, geroprotectors, this is what I was talking about, these things that maybe might increase the size of that sphere and keep it from leaking. So, examples of these are already evident, and maybe we can talk a few about them. We've been talking a bit about metabolic syndrome this morning, but not very much talking about the GLP-1 analogues. Maybe that's something that will come up in the discussion. But obesity or dealing with obesity could be something which could be thought of as a geroprotective approach. We haven't talked much about smoking and alcohol, and I do think that actually alcohol and smoking are both toxins which are affecting multiple organ systems at the same time. So stopping those could be conceived of as a geroprotective approach, and potentially the same could be said of air pollution. Physical activity, I'm going to show you a little bit of data about why I think this is a major geroprotective mechanism. And then we've talked quite a bit today and also yesterday about diet, but I'm going to talk a bit about the gut microbiome and aging and how I think that actually we can harness those as geroprotective mechanisms. But then there are lots of others. We've talked a little bit about some of the drugs already. There's also the possibility of young blood, young EVs even. Yesterday we heard from very interesting talk about taking EVs from stem cells in young individuals and how they might, well, they rejuvenate. Certainly they seem to rejuvenate mice. Humans, of course, the experiments are not done yet. I've been doing this for the last sort of 15 or so years by looking at twins. And of course, the reason why I was looking at twins is because I'm very interested in this intra-individual aging over time when you have got the same genetics. And so, of course, it's difficult to study one individual like that, but actually twins are a very useful natural experiment where certainly identical twins have the same genetics, but they may have different aging because they have different exposures across their life. And if we measure them carefully in a longitudinal study, can we find insights that are really helpful and actually capture maybe a little bit more of causative influences? So here, these are all identical twins within our cohort, and it's amazing to see how different they are. Because they share these genetic and early life environments, but they extensively don't share a lot of their adult environments and their exposures afterwards. And they don't share, certainly they don't share mortality to a degree, and they don't necessarily share all the diseases, certainly not at the time of onset of them. So when I started back sort of about 15 years ago, I was really interested in whether or not there was a single factor which might explain different biomarkers of different organ system functions. As I said, those brown lines that I showed in that diagram. So let's think about those brown lines. Let's try and map them to different organ systems. And we used an approach that was derived from medical literature to do that. So we looked for things that maybe ECG parameters and blood pressure parameters that could be able to describe the functioning of the cardiovascular system. The lungs, we took FEV1 and FEC and other measures of lung function. With the muscles, we took DEXA measurements. With the skeletal muscle, the skeleton, we looked at bone mineral density, but also some blood parameters of bone function. And metabolic function, we looked at those in similar ways that David has been talking about earlier on today. And we looked to see with a twin study are the factors. So we looked at these factors over time. That's the first thing to say. We looked at the development of these, the change of these over time. And we looked to say, are they changing together in all these individuals, or are they changing differently? And if they are changing, are the genetic and environmental factors that are underlying that variability in the change in those systems, are they the same across all the systems, or are they different across all the systems? And what we found was that they are very different across all the systems. We didn't find a very strong sharing of genetic factors that contribute to the changes in these organ functions over time. We found, in fact, very low shared heritability of the variance in change in these symptoms, but we did find shared environmental factors that then led to the changes of this organ system functioning over time. So different systems age differently. So remember, I've just put that sort of pizza of the biological pathways. These are happening differently in different organ systems, which is one of the reasons why things are complicated. But some of these environments are having pan-system effects. And so let's talk about some of these pan-system effects. So I was looking at this was actually my PhD. I was looking at what are the factors which are predictive of change in cognitive function in the twins over a 10-year period. And we used a wide range of cognitive tests using a computerized battery to really test their cognition underneath that ceiling level. So much more sensitive and specific tests than are used generally in my clinical practice. And we also looked at the brains of those individuals after the 10-year period. So look at structural changes in the brains. And I was really, as a clinician, expecting to see that cardiovascular factors or inflammatory factors indeed were the strong drivers, or even nutrition factors. But what I found was the really most strong driver was actually the strength of those individuals' legs back 10 years previously. And I think this is really interesting because I think that it's replicated in many other studies that show that physical fitness and physical activity, but physical fitness, number one, is really quite a strong driver of multiple domains, both multiple domains of cognition, but also whole body aging. And indeed, so here you can see two MRI scans of a pair of twins, and the twins have incredibly similar brains, but you don't need to be a neuroradiologist to see that the individual on the top has got much more white matter, much more grey matter, much less empty space. The ventricles within the brain in the middle, the black area, is much smaller than the twin who was much weaker 10 years before. And this was true across the population. So that's number one geroprotector that I might think to tell you about. But the thing is, how does this actually work on a biological level? And another study within King's College London, which is really interesting, has been looking at master athletes. And this is Norman Lazarus, who is a master athlete himself, who took up cycling actually pretty much around his retirement time. But he's now cycling still something like 200 miles every week. So they studied these master cyclists, and they've shown that the immune system is very much younger in these individuals than it is in age-matched other individuals. And so coming on to sort of thinking about, well, what do we do about this? You know, people know, don't they, about the geroprotective effect of physical activity. But what are we going to do about it? How can we get people to be much more physically active? And I was really interested to see a genetic analysis of UK Biobank data where they've taken wearables on these individuals and looked at their actual physical activity, and they've looked at what are the genes that are associated with actual physical activity and moving about. And of course, what they've seen is actually many of the genes that are related to physical activity are in the motivation system and neurological system. And so I think, you know, this is interesting and useful because there could be ways of harnessing that. And we have seen, again, with the GLP-1 analogs, which principally change motivation to eating, that they have major effects, potentially if there's a way of capturing motivation for movement, this could be a geroprotective. And I was really interested to hear the talk yesterday on EVs, because these mice that are given EVs, they were very different in terms of their function within the cage. They are moving around a lot more, and that's very interesting. So, coming on to another geroprotective mechanism that we've studied within the twins. Actually, because of that issue about how do we motivate people to move more, being sort of stumping me in my ability to take forward the physical activity research, I was really interested in the growing literature about the microbiota, because it's possible to modify it. And therefore, it could be a strategy that we should look at. So at that time, Paul O'Toole, who was a collaborator on this study as well, he'd published a really nice study in Nature, which was looking at older adults living in sort of rehabilitation or long-term hospital environments, sort of like a care home environment versus people free living. And he saw big differences in the gut microbiome, but it wasn't quite clear whether or not that was because of the environment that they were living in and the diet that they were then having, or whether it was really related to the frailty. So we looked at the frailty, the relationship between the frailty index and the gut microbiota in our twins, and they were all community-dwelling individuals, and we looked at their diet as well. And we saw that the robust twins had much higher numbers of this core microbiota that produce butyrate, in particular things like Faecalibacterium prausnitzii, so these really strong anaerobes which are very collaborative within the gut microbiota and support other microbes. And so the robust twins within the cohort had much more of these microbes than their sisters or brothers that had that were much frailer. And indeed, we know from inflammatory bowel disease studies that these same microbes appear to be causally helpful. So, for example, models using colitis in mice, induced colitis in mice, when they've got these microbes, they're able to recover better. And so we look to see, well, what are these things related to? This is two next papers just to take this further. So we looked actually to see whether or not these microbes are associated with disease. So thinking about Davide's discussion just now, and it was before your paper, Davide, so we didn't do a clustering approach, although we have done that since, and we replicate the same patterns as yourself. But we saw that this health-associated microbiome, led by these core microbiota like Faecalibacteria prausnitzii, was the same across multiple diseases. So it's not really about one disease. This is why you see in microbiota studies the same friendly folk popping up in association with things. And so importantly, we're thinking, well, okay, what is the key driver of this? And we're using the diet data we have within twins. We were able to see that this was strongly related to the plant fiber intake. And the particular species that, really, one of the species that's involved in this is Bifidobacteria species. And we know that there are certain probiotics which can boost these bugs. So then the next sort of stage of it was with Mary Neil-Loughlin here. We did a randomized control trial, a double-blind randomized control trial with maltodextrin as the placebo and the probiotic inulin and phos, these are complex carbohydrates that are derived from things like the Jerusalem artichoke, but also in chicory and other things. But they're quite low in the British diet, these things anyway. And we took individuals that were already quite frail. They already had low chair stance tests, and they did not have a very rich diet already. So in a sense, it was a targeted population. But we gave one of the twin pair that filled that study criteria, a protein supplement and this prebiotic, and one of the pair, the protein supplement and maltodextrin, and they were blind and randomized separately by a clinical trials unit. And indeed, we saw differences in cognition just in 12 weeks with this intervention. We didn't see differences in physical performance outcome because both groups really enhanced in their chair stands test after this. But we think that may be because there's not enough time for us to have really developed a change in muscles, but we did see changes in cognition in that time. And indeed, we saw changes in the microbiome. So here you can see a sort of little heat map showing which things changed across the prebiotic, which things changed across the placebo, and which things changed in both arms, possibly because of the protein supplement we gave. And you can see that those bifidobacteria were the things that increased indeed. Interestingly, when we gave this protein supplement, we did find that energy and fat intakes reduced in both arms, which was an interesting sort of like extra finding of that study. So ultimately, inulin and FOS does seem to make a difference in this short-term study, but of course, what's the clinical meaning of a change in cognitive performance on computerized tests? Does it really translate to any clinical differences? And so what we're planning now is really to think about interventions in clinical practice. And of course, when you're doing a trial, it's very difficult to do very long-term interventions. But older people, as I said before, undergo a lot of stresses in their life. And some of them we can predict when they're going to happen. For example, if they have a knee replacement for osteoarthritic surgery. And indeed, knee replacement for osteoarthritis surgery is quite a taxing thing to undergo. I don't know if anybody here has done that, but you have to really engage in very good physiotherapy to get over it well, and you have to do that quite quickly when there's quite a bit of pain around and so on. So it could be that actually giving a probiotic intervention could maybe enhance ability to get over that kind of stressor. So that's a trial that we're planning now. But we're also thinking about how this might affect other stressors as well. I just wanted to talk a little bit about another sort of area of geroprotections that we should probably be discussing in the panel, which is not just about what we eat, but also how we eat. And this study is a study that we did with twins before the COVID pandemic. We did it within about 1,000 twins, where we brought them into our clinic and we fasted, and we gave them a muffin which was made with lots of fat in it. And we had a look at what happened to their triglyceride rises and their glucose rises after we gave them the muffin. And there wasn't meant to be any people that had diabetes in this study, but as you can see from the bottom graph, there's definitely one individual who would be clinically diabetic. But we looked to see what explains the variability in their response to that food. And as I said, that these are actually normal individuals. And the summary sort of figure, I think, is quite interesting and quite thought-provoking, in that actually the meal content and the meal context was minimally sort of related to this change, but the meal context, as it were, whether or not they'd exercised beforehand and the sleep that they'd had beforehand and so on, seemed to be quite strongly driving the difference after their specified meal. But indeed, there's a huge amount of grey area here where the response to food, in terms of the things that we measured anyway, was not explained by all these other factors that we did measure. And so I think there's a lot more to be done in the context of meal. And obviously, others have done studies on the way that we eat across the day. But over the COVID pandemic, we did a very interesting study with something like 800,000 individuals doing an intermittent fasting, and showed some really interesting results, which we can discuss another time. But it's something that I think might be really interesting to explore further. In fact, that study was a study that was then led to a spin-out from King's College London called Zoe Global. You can see Zoe there in the bottom. And their interpretation was that we actually need to measure all these things very carefully, and we need to all have a very personalized diet because identical twins do have different responses. But I think that maybe we need to look harder about how we eat, and that might be more fruitful, and then maybe that we don't all need a personalized diet. So coming back to this intra-individual aging resilience and wanting to think a little bit about this middle one, number 2, which is the stressor. Obviously, the whole world went through a very significant stressor recently, and we were mapping that very carefully within our cohort, which is the COVID-19 pandemic. And we not only mapped whether people got infection or whether they changed their lifestyle, which obviously all older people in the UK were asked to change their lifestyle very significantly and stay indoors. But also many people changed their diet and changed a lot of their other habits. And we mapped these over time, and we also looked at the psychological stress of the COVID pandemic. And so we're going to be using this in the next, well, we're 2 years in to a very big project where we're going to be using all this information. And so as we've done over the last 30 years, we've been measuring lots of the biological measures that we've been talking about today. For example, inflammation, microbiome disturbance. We're also looking at the ways that proteins are spliced, splicing dysregulation and epigenetics, and altered metabolism, especially lipid metabolism. Over the time points before the pandemic, we've got our measures of pandemic exposures, including all these different types of exposures. And then now we're collecting the follow-up data, including that follow-up biological data in both individuals within the twin pair. And I'm going to show you a little bit of the results from this. And one of the reasons why we wanted to really look at this is because before the pandemic, we could see that there are differences in the trajectory of change in frailty, which is principally a composite of diseases and functions measured across, in our case, just about 40 different domains. And we can see that when we look at this in the whole population, it's very messy. And that's the reality of aging, is that it's very heterogeneous. But when we try and separate that into different clusters, we see that there are different types and different trajectories that people take. So in the group 1 in purple here, you can see that people are really quite rapidly changing over time. In group 2, these are people that already had quite a significant degree of frailty by this measure at the beginning, but then they stayed in the same bracket over time. And then there are people that seem to miraculously recover, which we think might be actually sort of error in measurement. And then there are people that we call normative agers, who start fairly low and can carry on, but don't dramatically increase. So looking at those things, are those going to be the things that then explain the change after the pandemic, and what happens to them later? And so this is this sort of like the basis of the data. And in that data, those data sets of those individuals before the pandemic, we were looking at inflammation, inflammatory factors, and I don't know whether well, I don't know whether the ones that you're interested in particular, I don't think they are, Davida, but we do have them in our sample. We were looking at whether or not inflammatory markers predict future frailty within this pre-pandemic set. This is unpublished data so far because we're waiting for our final dump of data. And there's not enough in this sample to really show really clear signal. But what you can see here is that when we look at factors which across the whole population, so these are the ones that are in light green, inflammatory factors are associated with change in frailty in individuals, when we consider them all as individuals, there's significantly more signal there. But when we look between pairs, only some of them seem to pop out. And that's what we were trying to do with this next study, is to see which of the factors, which even despite shared genetic factors and early life factors, are still going to influence trajectories of frailty. And the reason why we think that's important is because those factors might be more likely to be causal. And so here's another way of demonstrating that you can see that the things that are in the purple are things which you might do in a big analysis of lots of different individuals, and you actually find that certain proteins pop out, or protein clusters or groups of proteins indeed. But if you look within genetically identical individuals, we get a different set of signals. And one of the reasons why we were waiting before we publish this for further data is because we really want another data set to check that the signals are robust, because we think this is quite an interesting set of data. So what happened to those normative ages after the COVID-19 pandemic? And you can see that some of them really got a lot worse than was predicted by their trajectory beforehand, and some of them actually got a lot, seemed to be improved. And we're looking very carefully at what are the drivers of that. Is it things before the pandemic, or is it actually the stressor of the pandemic itself? And on the right-hand side, you can see a graph which is looking at grim age acceleration. So this is an epigenetic clock, looking at whether or not that epigenetic clock, a global measure of biological aging, stress within a system, whether it changed across the pandemic. And we saw that it did, but that was not related to whether they got infected with COVID. That was related to the stressor of the pandemic itself. And indeed, when we're coming to look at this information, we're going to be very interested in terms of whether it's these biological mechanisms that I've talked about, inflammation, metabolic dysfunction, microbiome alterations are the three that I'm really interested in, whether or not it's those things that drive this biological aging resilience measure and clinical aging resilience measure, or whether it's actually the pandemic stresses. And I was thinking that it was going to be more those mechanisms, but now the early data is coming out, and we're seeing that actually the size of the stressor might be really much more important. And that's really important in terms of where we go forward with aging research. I've talked to you about how our body systems age at different rates. Maybe there's lots of future me's, different sorts of future me's. Different bits of us are aging at different rates. We may need different approaches for different systems, which could be personalized for ourselves, or we could be focusing on finding those pluripotent geroprotectors. And I just wanted to remind you that where there are shared drivers, these are drivers that are generally environmental factors, and they are acting across the life course. And just to sort of sum up, especially thinking about those stresses that we're increasingly interested in looking at in individuals as they age. And we're coming to quite troublesome time over the next sort of 10 years. We're going to be seeing increasing numbers of stresses. And I'm really pleased that Jan is talking tomorrow about some of the environmental stresses coming from climate change. But there are other sorts of environmental change that are happening, which are sort of forming a perfect storm with an aging populations. But could there be common solutions? And this is where I think we need to urgently come together interdisciplinarily, from interdisciplinary perspective, to work on new strategies to promote healthspan, to lessen the effect of these stresses, but also increase resilience within populations. So within King's, we're bringing together a network of researchers that are working all the way across all of these different themes, including people from geography and social science, population health and biology, to study aging. And we've already had some success. I won't delve into this, but going all the way from really looking at the biology of a stressor like colorectal cancer, which Carly Welsh is leading to, all the way through to how we deal with pathways of care for older people undergoing significant stressors like surgery. I'm going to sum up with a few conclusions here. I think it's important to consider what we're trying to do amidst the complexity of studying aging. I think that it's really important to be thinking about the fact that we're here to increase health span, but not necessarily to increase lifespan. I think that the key focus really could be on geroprotectors which have pluripotent effect. And I've highlighted again, and as other speakers have done, the importance of physical exercise, nutrition, and like Eli Metchnikoff might have predicted 100 years ago, the microbiome, as things that we can easily change now. But there's a real opportunity for new geroprotective drugs as well. But I think it's also important to consider the stresses that older people go through in life, because I think actually those things then cause tipping points, and we need to be thinking about how we lessen them for the most vulnerable. And I just wanted to give a great thanks to Michele Ferrero, who had huge foresight in focusing on active aging, and that the urgency for developing new strategies for health span is ever more apparent. And there, that's just a photograph of some of our twins on a party that we recently had. And finally, I'll leave you with a picture of a carer of one of my patients with dementia, who is 94 herself and as fit as a flea. So it is possible. Thank you.
Thank you very much, Claire, for this very interesting data. And we open the discussion from the audience. Question from Davide Vetrano.
Thanks, Claire, for this great talk. I noticed that when you asked AI to draw 2 pictures of you, when AI drew the picture with a poorer prognosis, it drew you with like a bit more gray hair, and it put you like a darker sweater, which reminded me about the importance of like always considering what is a confounder, darker sweater, when you observe these things, and what is like just a proxy, the gray hair. Speaking about proxy, and you mentioned geroprotectors, and you are planning also intervention studies for like improving healthy aging and so on, and health span. What would be your favorite proxy or indicator or measure of efficacy of in these trials?
Yeah, so yeah, this is a very good question, and I think so the question is, what should be our outcome measure in trials for healthy aging? I think that's the answer, and this is a super important question, which we have to grapple with in multiple different ways depending on the setting. So ultimately, we know what we want to do is we want to have people living functionally happy lives, as it were, so mental and physical health and well-being, ability to function, and not using healthcare services. Yeah, the trouble is to do trials in that sort of like magnitude for those things. You would have to do very large trials. So we need to find biomarkers which measure these functional factors more closely. And so that's where things like gait speed, is a nice composite marker, and we know what might be a clinically significant difference in something like gait speed. We know that, cognitive markers, we know some of the thresholds that are clinically important with cognition. So we can use these other markers. The thing is that actually, for some of the interventions we want to study, for example, in heat stress events, reducing heat stress events, we might need specific biomarkers which we know change in the context of that specific stressor. And so one of the studies we've been doing over the summer is actually putting in a whole load of tech into people's houses in London and measuring what are the things that change and are associated with those other more important functional markers, which we could use as end points in a trial to then study some of these other geroprotective agents within the context of that specific stressor, because those markers have to be sensitive to the stressor. And that's why it kind of depends on the study.
Yes, there's another question from the audience, and I think the last one, and then we will move on.
We make some huge assumptions as to what really stressors are, and we talk about COVID-19 being the ultimate stressor. I actually had a good time. I enjoyed the 3 months at home. I had, got to learn to cook really well.
Yeah.
We now live, I think, in a much more stressful situation in the United States than we did during COVID. Same thing with Ukraine, same thing with Russia. Do you think that this, in fact, is, the other thing is I found that hip replacement, which I actually enjoyed having because I could go back to my 10K running, was actually a positive aspect of my life. So how do we assess what really is a stressor? Can we ever really say this was a stressor or not? The other question that I have is the assumptions we make. For example, GLP-1s, I actually think they may be putting us at a great risk. Because first of all, you've got this incredible degree of, let's just call it atrophy, muscle and adipose tissue. You've also got a miscommunication of your entire limbic system, your reward system, your motivational system, your attention systems. That's why you don't want to have a cigarette. That's why you don't want to take drugs. That's why you don't want to eat. But that's also maybe why you don't want to go to work. And you may not want to go out and have a job. You may not do it. So how do we interpret all this complexity? And the last thing I just want to say is I admire your quagmire of data and how you swim through it and actually generate some very interesting testable hypotheses.
Well, those are three fantastic questions. I love the question about the stressor because that's exactly what sort of we found within COVID pandemic is some people actually really thrived in it and actually got better. And some people didn't. And we measured very carefully what happened to their diet, what happened to their physical exercise, what happened to their mental health. And interestingly, actually, older people in general reported much better, relative to younger people. Basically, COVID was terrible for their mental health. Older people, actually, very much less so. Really interesting. And that's replicated across countries across Europe. So I agree with you that, let's not think of. Well, I mean, COVID-19 undoubtedly stressed our systems, but it did different things for different people, and that's why it's so interesting to look at. But yeah, we need a big study to do that because there's a lot of variability in that stressor. And very interesting about your hip replacement as well. But I imagine, when we think of that as a stressor, we might be thinking of the short-term effect of the surgery, which then, of course, you got over and you did really well afterwards. But your next question with GLP-1 analogs, I think we're going to be talking about that later on today, and I think I'd like to enter a discussion about what we think about singular drugs, and also what do we think about singular drugs which we might use for a short period, and then what happens if we stop them, and then suddenly we've lost muscle mass, we then put back on the weight, we might generate a whole nations of sarcopenic obese individuals. So there are lots of things to think about here and study here, and we need to be working together as a community to study them.
Thank you. Passiamo al prossimo speaker della